happy to introduce Marshall Porterfield. He is a professor of biological engineering at Purdue, where his research focuses on biosystems and the space flight environment for development of biomedical countermeasures and bioregenerative life support technologies. He recently served NASA as a division director for space life and physical sciences, including human research and space biology programs, focused on science for future human exploration using the International Space Station. During his tenure, he developed a new open science paradigm for the utilization of the ISS. Today, he will present a talk titled Hacking Life, Engineering Biological Systems Beyond Genomics. Please take a moment to silence all electronic devices, but feel free to tweet using hashtag Dawn or Doom. Thank you and enjoy.
Okay, now you can hear me. Probably didn't need it anyway. I'm also going to talk about how the emergence of the ability to reprogram living systems at a really fundamental level, way beyond what we're doing right now with gene editing. That's really where we're going to be going. And we're going to talk about how that can be applied to sustainability and building on what we're doing with genetically modified organisms for agriculture and medicine. Specifically looking at human medicine and the potential of these technologies, and also looking at how they're going to play an important role in extending and creating new capabilities for human space exploration, habitation, colonization. And I also want to point out here that when I say hacking life, I really do mean reprogramming how biological systems work. We're not just cutting and splicing genes anymore. We're going to be able to interface with the genetic systems at a sufficient level to really understand the operating system. So I'm not talking about biohacking. I'm not talking about which supplements you can take to make you remember things better in the classroom or get six-pack abs. I'm talking about reinventing life. So in order to start off this journey, we're going to take a look at where we've come from and take a look at where we're going to go. That's going to be kind of the theme. Starting off with the Human Genome Initiative, about a 10-year project, over $3 billion, in several international teams, over time chopped up and sequenced and reassembled a human genome. It was actually one of the challenges they faced was really how do you deal with that the amount of information. So at that time, 3 billion base pairs, that amount of data was big data. It's nothing now, right? We have thumb drives that by far exceed that. But at the time, that was a big deal. And so the biologists started collaborating with information scientists. And not only did we get the human genome, but we also got a whole new field of bioinformatics. That gave rise to medical informatics and the promise of predictive medicine. Take a look at your genome, figure out what diseases you may have, which drugs may work for you, and actually be able to treat you in that way. But that promise did not come through. How many of us today go to the doctor's office and they pull up your genome and take a look at it and figure out what drugs you should take? That hasn't happened yet. But that at the time, in 2000, there was a lot of predictions of that type related to the outcomes of having the roadmap, the genetic blueprint for human beings. Well, now we can solve all of our medical problems. It hasn't happened yet. Why not? Let's take a look at the complexity of the system and look at in order to understand this, we need to understand the flow of biological information. So the genome is just the coding. In order to understand how the genome works, we have to understand how biological information is transformed from the genome into biological activity. And there's several steps along the way. We have the genome and the field of bioinformatics. We have parts of the genome which code for genes. So Mendelian genetics. The coding of genes goes through RNA. The RNA is transcribed, it had the transcriptome, into proteins. So if you look at the collective content of protein in a cell, that's called the proteome. And we have technologies beyond genomics to measure and study these different types of omics components of living systems and follow the flow of biological information. In biology, proteins, everything is made of or made by proteins. Proteins do everything, really. And to make this whole thing more complicated, if you look at this flow of biological information through biochemical pathways to the metabolome, the ion contents in the cell, the ionome, all of this stuff is talking to itself and talking back to the genome. There's this flow of information that's absolutely incredible in this system. And we're going to look at the complexity of that from a computer science perspective. Now, the human genome, they started mapping it, and they discovered there's different estimates for the number of genes, from 19,000 to 24,000 genes that takes to make a human. All right? That's only about 1 to 2% of the genome. What the hell is the rest of it doing? Right? Only 1 to 2% are proteins. Only 1 to 2% we can actually look at and say, okay, there's a functional thing in the nucleus of the cell, in this control system. We have absolutely no idea, or very little idea, only an emerging idea of how it all is processed and controlled. So, as a more simplified view, this is the central dogma of biology. DNA contains genes. Those genes are transcribed into RNA, and RNA is transcribed into proteins. If you look at the technologies for analyzing the DNA, that's genomics. The technology for analyzing RNA, that's transcriptomics. And the technology for analyzing proteins, that's proteomics. These different collections of omics, what we're trying to do now is start to integrate those and look at the relationship between the genome, the transcriptome, the proteome, and other aspects of biological activity. It is a big data problem, okay? If we have 20,000 genes, that may give rise, because of there's gene editing and different types of processes for modifying the information from the genes before they're converted into proteins. We may have 40 to 50,000 different types of messages, and maybe 70, 80,000 different types of proteins and protein complexes, and hundreds of thousands of different molecules in the metabolome too. So if you look at scaling of just these three elements, if you took one sample, one cell, and analyzed it for these different omics channels and combined it all, and looked at it in terms of its data complexity from a systems level, that's about 10 terabytes of data, of raw data, per sample, per second. This is big data on steroids. When people talk about big data, this fulfills all of the 4 V's of what we think of as defines big data. Volume, velocity, variety, and veracity. Veracity reflects the complexity of the interrelationship of the elements in the data. And that's really where biological systems greatly exceed what we're playing around with in terms of Facebook and Amazon and Google and the World Wide Web. Biological systems really represent the ultimate big data challenge. So for you computer scientists out there who are looking for something to do in the future, you might want to think about this. So, and I like to look at this from the perspective of analogs of computing. So if you look at the genome, that's just really the hard drive, right? The hard drive, including the coding for the operating system. This includes driver programs and the kernel, right? Genes are transcribed into messenger RNA. That's when elements from the hard drive are moving up into RAM, and the programs are being accessed by the processor. And then the proteins themselves are really just like the application programs. They're individual programs that have individual activities, and they go out in the cell and run their own little program. And then the metabolites are like I/O devices. So the idea is to take a kind of reverse engineering perspective from a computer science perspective and combine that with integrated omics in order to create a model, ability to emulate what cells do. To understand this system, it's with such a high level of fidelity that we can use that type of information for next generation engineering. Really engineering biological systems from the operating system level and from the processor level. So this is a dramatic change from what we're doing now when we're talking about gene editing and gene splicing types of approaches. So let's take a look at some examples. We're going to focus on sustainability and take a look at what we can do with GMOs now and what that might mean in the future. We're going to take a look at what we're doing in medicine using gene splicing and gene modification approaches, and think about what we can do if we can really change the programming for the human genome. And also take a look at how this applies to the future space exploration and what we're doing on the International Space Station today. So here we are. This is another way of looking at genes as components using a kind of a computer science perspective. If single cells are computers and we start to scale up to organs and tissue systems and whole organisms, now we're talking about networks and webs of information. So again, we're at another level of complexity. We start trying to scale up to that level, and you start thinking about systems biology transitioning into synthetic biology, species engineering, ecosystem engineering from this, using this type of approach. And you can think about how far off is this. If you consider the fact that Moore's law is held up for processing, we may not have the data processing capability to hack, to create the emulations of living systems today, but that's coming. Artificial intelligence may contribute to this. Actually, this may contribute to artificial intelligence. Machine learning is definitely going to play a role in this. But if we can emulate living systems at this level, and we usher in this new synthetic biology era where we can really reprogram living systems at a fundamental level, it's going to have profound implications on how the planet works. Now, right now, we're already terraforming the planet. We're already changing the atmosphere. We're changing the ecosystem. If you take a drive from Chicago out to Iowa, you will literally drive through hundreds and hundreds and hundreds of miles of GMO-modified crops. The ecosystem is not a natural ecosystem. It's what we make it right now. Now, we have the choice to make it good, or we can let it just degrade. And at some point, we're going to have to figure out how we're going to recover a planetary ecosystem. And are we going to walk away from these capabilities? Or are we just going to let things just kind of flow the way they are right now? Are we going to take control or let it happen to us? So I think we are ushering in a new synthetic biology era. I think we're looking at if Moore's law holds up for computing, if Moore's law holds up for omics technologies, genomics, the $100 genome is coming. 23andMe is, lots of people have 23andMe accounts, right? That's not true genomic sequencing, but the $100 genome is coming. And so these technologies are keeping up with Moore's law, and when they combine and collide, we're going to be ushering in a new era. Estimates, 5 to 10 years from now. Things are happening fast. So in order to kind of get an idea of where we're going to go, let's take a look at where we're at now. A lot of controversy about GMOs in the environment, agriculture. I could do a whole talk on GMOs, obviously, so we're not going to spend that much time on it. It's politically relevant. The NAFTA agreement is being phased out, and the next generation agreement is actually addresses genetically modified crops and trade between Canada, Mexico, and the United States. A lot of controversy about whether GMOs are safe. In theory, they're safe, but how they're implemented might not be safe. So there's always a caveat. Now, when people come out and they flatly say that they're against GMOs, I think it's important to point out that GMO insulin saves a lot of people's lives every year. Right? So GMOs are not just bad crops. They're actually platforms for producing biological proteins that can be used for drugs to treat human disease. So GMOs are not necessarily, all have the complete potential for causing problems. But we have seen problems with some of the GMOs. And it's not necessarily in the GMO themselves, it's potentially in how they're used. So Roundup ready soybeans, essentially most of the North American soybean crop now is Roundup ready. It's genetically modified, has a resistant gene that they discover from a weed. It's the same gene that's in the soybean. They just put a different version of the gene in the soybean. Now you can spray Roundup on your crops as much as you want to all day long. Your soybeans are going to grow, but you're putting a lot of herbicide in the environment. So while it's been a financial success for Monsanto, and the farmers are willing to pay the price for it because it increases their productivity, it is associated with overuse of the herbicide and actually increases the use of the herbicide in the environment. And now we see Roundup is showing up in our food supply. Reports over the last few weeks have suggested that Roundup is in our cereal now. Another example is Bto Corn. There's a protein from a bacterial species, Bacillus, that disrupts digestion in caterpillars. So they transform the corn with the Bt, and to some extent, this really is associated with about 85% decrease in pesticide use. So you don't have to spray the pesticides, it's safer for the environment, but there's still concern about what's the ecological impact to other species in the ecosystem because of this, the pollen and other runoff. I mean, it's not like there's a little bit of corn growing out there. It's growing everywhere. It's the entire state almost, right? So we're talking about a lot of transgenic proteins floating around out there. So what's the potential if we can really change the game and instead of gene hacking, basically operating system hack biological systems, the potential is that we can really create a lot of new opportunities for crop development. Already we're using almost all of the land on the planet that's possible to be used for agriculture. So it's going to be important as the population grows, 2050, another doubling of the human population, that we can increase productivity on the land we already have, but can we use synthetic biology approaches to create new crops that can grow in arid land, soil with lots of salt. There's lots of other environments and other potentially unusable land right now for agriculture that we could open up through really wholesale reprogramming of plant systems for agricultural productivity. But just like any other technology, I think it's really important that we evaluate what we're doing and be careful about what we're doing in the environment. We're already doing a lot of things in the environment, so let's not pretend like we should never do this because it's already being done and we're already causing damage. Can we improve things? Can we improve the human condition in a safe way? So what about gene splicing and gene therapy in humans? There's been a lot of press recently about a new approach for treating cancer. They harvest your immune cells. They genetically modify them to trick your immune cells so that they know how to sniff out the cancer cell and kill it. So it tricks your own immune system. And now you are a genetically modified organism. How many people that are against GMOs are going to turn down life-saving therapy if they have cancer because they're afraid of becoming a GMO? Because that's essentially what gene therapy is. You as an individual are going to become a GMO. There's a lot more coming. Treatment for blindness. CRISPR is a biotechnology that allows for precision in gene splicing, introducing new genes. So again, we're just talking about gene level engineering, not genomic computing level engineering, not reprogramming life. The U.S. intelligence community has identified CRISPR as a potential weapon of mass destruction because of the impacts of what you can do in terms of bioweaponry and bioterrorism. The 2018 Nobel Prize was awarded for the work that led to the new advances in gene therapy as it's applied to cancer. So these are things that are going on right now. What's going to happen in the future when we have expanded capability to reprogram the operating system, to rewrite the processor for human beings? Even today, with the technology that exists today, it might be possible in the next 5 years. It probably will be possible to actually be immunized against cancer. If this works, they can go in and harvest your immune cells, identify 10 different types of cancer, modify your immune cells for that, and you walk out vaccinated against cancer. 40% of us are going to face cancer in our lifetime. Imagine we just wiped out in 5, 10 years. Just using the technologies available today, we wiped out cancer. Life expectancies would jump up to 100, 110 years almost immediately. What about beyond that? What about systems level reprogramming of biological potential? Are people going to turn down vaccinating their children for cancer if you're anti-vax? So there's a lot of social implications for this. There's a lot of ethical considerations when we start moving beyond just gene level editing of you as an individual versus doing things that are going to change the germ line. Right? Writing in permanent changes to your genome in such a way that is passed on to your offspring. Already you can go to an IVF clinic and receive treatment for infertility and select the sex of your child. That's already being done. What happens if you can select your eye color and other traits? We're already doing experiments. We've already shown that we can do this in embryos. Scientists in China and just recently in the United States have been replicated. Using CRISPR and other technologies, you can rewrite at the gene level the genome of embryos. And this is permanent in the species. Already doing work with other animal systems that can be applied to humans. So what if we can re-engineer what it is to be a human being? What if we can re-engineer our capacity to cure cancer? What if we can re-engineer other major diseases? Perhaps 20 years from now, the freshman class that enter Purdue will have lifespan expectancies on averaging 200 years. What if you can do regenerative medicine? What if you can reprogram? So we're programmed to die. Cells are programmed to die. We're programmed to have a lifespan because it's part of evolution. In order for the species to evolve, you need reproduction and recombination every generation. What if we outgrow that? What if we have lifespans that exceed our reproductive capability? I think there's a TV show that has to deal with that topic, right? What if this is a future that can happen in 20 or 30 years? These are all potential things that are the result of the whole cell ability to reprogram life. This is really deity-level dominion over the control of human evolution. Deity-level dominion over the control of life on this planet. In a short period of time, combine omics, combine it with AI, create these new program-level pictures of how living systems work. That's the type of power that we'll have. So are all of our children going to be Olympic athletes with Einstein-level intelligence? Is it going to be a utopia? Or are we going to struggle along the way and somehow this gets subverted and used to develop the super soldier of the future? That's our choice. Dawn or doom, right? Why choose dawn? I hope that we're on the verge of using these types of technologies to do good things. I hope that we'll realize as a species that we really do need to embrace the idea of becoming a multiplanetary species for the sake of human evolution. If you just look at human population and the finite resources on the planet and the rate at which we're degrading it, it really is important that we identify a pathway forward for human civilization to continue to grow. Because we can't really stagnate, we can't really all become, we can't eradicate death and just have a fixed population of human beings on the planet forever, the same people. It just doesn't, that's not an evolving species. That's not a viable species in the long run. So what are we doing in space? Well, really you don't have to look any further than to the International Space Station today to see that integrated omics is actually really an important part of our space program. The research that's being done on the International Space Station is really important just to take the next steps. The journey to Mars, Mars exploration, even a very simple mission to Mars. Right now today, from a biomedical perspective, is not possible. I know a lot of people don't say this. I spent 5 years at NASA headquarters. I was in charge of the human research program, fundamental space biology, and the biomedical issues just associated with the microgravity exposure would be detrimental to crew if we just use the existing technology. If we use moon mission type of mission architecture to go to Mars today, they would get there and probably wouldn't even be able to walk around in the Martian gravity environment. That's without considering high energy galactic radiation and the radiation environment beyond the low Earth orbit of planet Earth. They would probably arrive very sick from radiation sickness or with cancer and probably in the state of dying. We don't have countermeasures for it, so it's really important that we use the International Space Station to advance the research, the biomedical research, and the human research that's necessary to develop the next generation technologies for human exploration. And Mars is just the next destination, I hope. So what are we doing on the International Space Station? So around 2012, we started developing a plan, and basically it fully embraced the idea of using integrated omics, going genomics and beyond, and collecting large integrated data sets associated with living systems and microgravity. These are referred to as reference experiments because we're not doing science based on individual hypothesis like we had done in the past. Really using a 19th century model for managing science on the International Space Station. We get, for rodent research, we get 2 experiments a year. That's not a lot of throughput if you're going to solve the medical problems that are associated with every aspect of the human body. The neurological system, bone, muscle, cardiac, immune, etc. Vision. The human body, and just in the microgravity environment, without the radiation, is breaking down and aging at a rate that's probably about 10 times quicker than what humans age at on the ground. So in order to address this issue with throughput, we're moving away from single investigators, single hypothesis-driven experiments, reference experiments which capture large integrated omics data sets, putting them in the GeneLab data archives, making that available to the broader research community, and issuing grants against that data so that people will go in and mine the data and analyze it in order to identify targets that will allow us to translate that knowledge and that data into new capabilities and technologies for human exploration architectures. And how many of you are familiar with the twin study? The one-year mission with Mark and Scott Kelly. There's an really interesting story about how this came about. But suffice it to say, after we negotiated the one-year mission with Roscosmos, we came back, handed off astronaut selection to the astronaut office and crew selection, and they came back with Scott Kelly. Now, immediately, we hadn't really taken advantage of the fact that he has a twin brother. But immediately, we identified the potential to do genetically paired twins, to do a research project starting off 1 year before launch, and then you have 1 year during space, and look at the body's recovery 1 year after. So there's really a 3-year window that that these 2 guys volunteered to be test subjects for. So they're actually the lab rats. And the approach that we took was really based on integrated omics. So we put out a research announcement. We selected 10 proposals. These are all related to proteomic, transcriptomic, genomic, all different aspects that we felt like are going to be integratable data in order to develop a integrated model of data that could be accessed 10, 15, for the next 20 years in order to be mined and identify targets for biomedical countermeasures. The first summary paper is going to be coming out this spring, I'm told. This should be the first big paper coming out from this. This will be an area that I'm going to cover with a lot of detail. If you're interested in this, you're an undergraduate student, I think there's a few slots open in my space biology and medicine course coming up this spring. So if you're interested in this and you're not aware of that course, there's my plug for my course. But we're really looking forward to the data coming from that experiment. Now, when Scott was on orbit, not only was he a test subject, he also conducted a lot of experiments. He was actually primarily responsible for the veggie experiment. In this experiment, he was growing different types of plants, and they actually grew and consumed the first crop of lettuce in space that was grown just for crew consumption. And so it's actually 20 or 30 years worth of research went into developing the hardware and the countermeasures to actually grow the plants normally in microgravity. And they benefited from that and really enjoyed the fresh lettuce experience. We did also provide some fresh balsamic vinaigrette to go along with the greens, and we understand they enjoyed that quite a bit. But why are we doing this? Why are we growing plants in space? If you look at any serious architecture, mission architecture, for a sustainable mission, whether it's an exploration mission to the moon or Mars, or for interplanetary colonization, a major component of the mission architecture is going to be agricultural plant-based systems. And that is the foundation for bioregenerative life support. So plants can recycle the atmosphere, they can purify water, they can recycle nutrients, they interact with microbial systems. There's a whole microbiome associated with plants and plant agriculture that can be engineered as part of a bioregenerative capability. Now think about it. That's what we need here on the planet, isn't it? We need bioregenerative capabilities to kind of maybe regenerate our planet a little bit from the damage that we've already caused. So what this means is that the same technology we need to get off the planet is the same technology we need to stay on the planet. But it's still at the same time, human population is growing, and humanity deserves a future, and civilization deserves an opportunity to grow beyond a single planet species. We want to be an interplanetary species, starting off in the solar system. If these synthetic biology approaches are going to be used to engineer these types of bioregenerative capabilities, and at the same time re-engineer human beings to promote and cure disease and regenerative medicine and minimize aging, that we're going to have to create new opportunities for people to expand civilization. At the same time, we're in a difficult period where there's a lot of friction, and there's always the chance that in the transition and aiming for a soft landing, that these types of technologies and synthetic biology approaches could be applied for warfare, or they can just be misapplied in a way that could have disastrous effects. So we really want to be careful with regard to that, because the future we want is one that ensures the survival of our species. That's really where we're at today. And it's really interesting now to see how all this technology is coming together and creating completely new capabilities beyond what we've ever imagined. I think that the idea of synthetic biology is really not that far off in the future. I hope that I've been able to convince you that this is something that's going to be coming in our lifetimes that we need to be aware of and have on our radar screen. I could talk a lot more about this, but I think I'm getting close to running out of time. At least leave 10 minutes for questions. Thank you all for your attention, and I'll be happy to answer questions now.
Thank you for the talk. That was inspiring. As a computer scientist, you mentioned computer science a couple of times. What are some of the skills that we need to have so that we can contribute to all the research that you mentioned?
I think it's really the main skill right now would be the ability to collaborate. And to some extent that sounds simple, but it's really not simple. In my experience, collaboration, if you get an engineer and a scientist in a room together, they can talk to each other all day unless one of them climbs up on the fence between the disciplines and decides to learn a little bit about the definitions and how to speak the other one's language, becomes bilingual across disciplines. The collaboration is not really going to work. So bioinformatics is an emerging and growing field. Bioinformatics and medical informatics are going to be changed by advances both on the big data side, but also on the biology side. So I think really what I would suggest is if you're a computer scientist and you want to play in this new domain, learn about biology. And if you're a biologist and you want to play in this new domain, learn about computer and information scientists. And find someone that's your counterpart and collaborate with them.
How much is hard science and how much is imagination?
It's all hard science.
No imagination required?
I would say extension of what is already happening.
Do you have any specific thoughts, ideas of how we will manage to reverse engineer the operating system, as you told earlier?
That's an interesting question. But unless we have the picture and we can identify the genetic elements as they relate to the processor and the operating system, once we can identify those, it's a matter of using CRISPR or some other just to rewrite the code. And looking at diversity of biological systems. Genomes are not a big deal anymore. After the human genome, people started sequencing genomes of everything they could find. Fungi, plants, mice, genetically modified. There's thousands of different types of knockout mice, genetically modified mice. Each one has a different gene knocked out of it. So we use those as models for pharmaceutical drug discovery. So all that information, what we learn from what we can glean from the operating system of the other animals, and how really comparative molecular biology is going to allow us to learn how to see differences in how different operating systems work, identify the targets, and really, it's in the genome. That's the funny thing is, it's all hiding right there. It's all right in front of us. But we have no clue. We have no way to tap in. There's no way to go in with electrodes and tap into the wires and listen to the electrical signals associated with how the processor is working. We just don't have that capability right now because we don't know where to look. And that's why it's really important to integrate these omics so we can start to use machine learning, artificial intelligence, and those types of data tools to emulate what's going on so we can learn how to emulate how it works and then hopefully identify how that relates to the actual genetic code. But how do you reprogram a processor or operating system in computer science? I mean, if you know where the code is and you can access it, but right now we really don't know where it is.
Excellent, excellent lecture so far. Question though, does NASA work with like big companies like IBM or universities to fulfill some of this data, or to, I should say, analyze this data that you guys have done? Or is it all, I mean, is it?
Yeah, we had talked to I'm not sure where the GeneLab project kind of landed with our proposed collaboration with Google, because it's GeneLab. The GeneLab program is based out of NASA Ames, and basically Google campus took over half of what used to be NASA Ames Research Center. So they're like right next to each other.
I was just wondering how, if the information, everybody sharing the information among, I mean, or is this top secret? It could be top secret.
Well, GeneLab is completely open. It's open science. It's open to anyone. So anybody with a computer science background or a bioinformatics background who wants to become a space biologist, all you do is download the data and start playing.
Start playing with logarithms and everything else.
Yeah.
All right.
But we talked to, we worked closely with the Broad Institute in developing the GeneLab platform. Leroy Hood with the Bioinformatics Institute, he was also an integral player in that too.
I was just wondering if like Purdue would be involved in something in a study that they would use or break down the numbers that you would have.
Yeah, there's regularly NASA NRAs that are providing opportunities for GeneLab analytics. So any professor here at Purdue could respond to those types of opportunities and get involved.
Okay, just curious.
Funded, yeah.
Thank you very much.
You talked a little bit about microphones. I feel like I should sing or something, but you talked a little bit about curing things like cancer and other diseases and illnesses. My question with that, we come up with a cure, boom, here, cancer cure. How would something like that be administered? Is that a shot? Is that an IV? Is that some type of procedure you would have to have done? Or have we even gone to that development stage yet?
In terms of the application of gene therapies right now, today, in terms of like in the clinic, it depends on the type of condition. Like with the issue with where they've been able to cure blindness, they actually inject CRISPR and related genes to correct the damaged gene right into the eye. And there are ways to trick the cells to take up the DNA and the CRISPR components, and it just really finds its way into the nucleus. So it's really an injection. With the immune system modifications, that's really, it's inoculating your bone marrow stem cells. So they harvest some of your bone marrow, they identify precursors cells, stem cells that are associated with the immune system, genetically modify them, and put them back in you. So now you're a genetically modified human walking around with a genetically modified immune system to kill cancer.
Okay, and then a second question that I had, you were talking about people living to the year, let's say 200 or beyond. My question with that then, we're working with kind of an allocation of changing the aging process as well, correct? Because I mean, most of the people that we see nowadays that are 100 years old, they can't get up, they can't move around. I wouldn't want to live like that to the age of 200. So we'd have to totally transform the whole person, including muscular, including thought processes, etc., in order to keep them productive up through the ages of 200 and beyond.
Yeah, there's we are genetically, I was trying to refer to that when I talked about regenerative medicine, reversing aging. The idea that we can basically reprogram biological systems to turn off that program within our genome. So that's really what regenerative medicine is. It's about reversing the effects of aging and identifying therapies, or even preventative medicine can be considered to be regenerative medicine, because if you prevent the disease from happening to begin with, the associated damage and the aging associated artifacts with that, but those are if you look at different disease models and how different disease models are associated with, say, reactive oxygen species and oxidative damage in biological systems, that's another target. They've shown that they can increase the lifespan and reverse and reduce the accumulation of aging effects in some model biomedical systems just by overexpressing and controlling antioxidant systems. So there are different components that can be identified today that are anti-aging. But I really think it comes down to reprogramming the operating system and reprogramming how basically telomere length on the ends of the chromosomes, that's associated with aging. So it may be possible that those actually may be a direct target for modifications. That's one of the differences between Scott and Mark Kelly when they came back, was differences in their telomere lengths. So Scott aged a little bit faster than his brother, but he also came back taller, so which really bothered Scott.
You talked about the promise of life hacking, but you also talked about needing to be careful in how we apply the technology. How do you see that getting governed? Who would govern the application of the technology? How do ethics and morality get applied to the application of it?
Yeah, that's a difficult issue. If you look at the early days of the molecular biology, the advent of molecular biology and biotechnology, there was a lot of concern by scientists, and scientists were involved with policy and ethics in developing protocols. That's why we have a lot of the protocols for human subjects, animal subjects, because concerned scientists have always been really proactive about identifying where there are risks and dangers and trying to minimize the impact of that on society and really on themselves too. It's kind of self-preservation for the scientific community to ensure that there's responsible use and responsible application. Now, having said that, that doesn't mean that DARPA doesn't already have programs in place where they're identifying genes that could be modified to enhance warfighter performance. The Army Research Office has a whole human performance division, and they do a lot of really interesting research looking at various aspects of physical and mental performance in human subjects. And part of what they're extending that to is gene level types of therapies, and even considering beyond that. So it doesn't mean that we're not going to do research and look at the possibility of these things and potentially identify that as this is where our risk is, and we have to figure out some way to make sure that the other guys don't get. That landscape of national security and defense that's a really complicated one, kind of scary too.
So I think we're at time. Could you stick around for a couple minutes up front?
Sure, I'll be happy to.
Okay, let's give him another round.